everybody. I'm Maggie Flanagan, one of the newish neuropathologists for anybody who doesn't already recognize me, and I'm very excited to host Dr. Lon White today. So Dr. Lon White graduated from the University of Washington School of Medicine and completed a residency in pediatrics at the same institution. He then received a master's in public health and epidemiology from the Johns Hopkins School of Hygiene and Public Health and spent 3 decades as a scientist at the National Institute of Health in Bethesda. As the founding intramural neuroepidemiologist at the National Institute on Aging, he established the East Boston Dementia Study, the Framingham Dementia Study, and the Honolulu Asia Aging Study, or HAAS. In Hawaii, he designed and oversaw HAAS operations from 1991 through completion of data collection in 2012. The accrued HAAS data includes 8 cycles of longitudinal clinical assessments of 3,734 participants and comprehensive brain autopsies on 852 decedents. He is currently the principal investigator on 4 grants analyzing the accrued data from the HAAS and the NUN study to better understand the pathogenesis of late-life cognitive and motor impairments with a special focus on multiple interacting brain abnormalities responsible for such impairments. Future development of effective prevention and treatment interventions for aging-related cognitive and motor decline will depend upon our understanding of these complex underlying pathogenic mechanisms and why some individuals are at either an increased or diminished risk for their development and clinical manifestations. So Lon is an amazing epidemiologist and very inspirational. He's kind of guided my interests in research, and yeah, I'm really excited to have him speak today on progress toward understanding the pathogenesis of late-life dementia. So thanks, Lon.
Thank you very much, Maggie. It's a delight to be here. It's a lot colder than what I'm used to, but it's a delight to be here. What I'd like to do today is sort of tell a story, and the story really is about how central the discipline of pathology is to our understanding of Alzheimer's disease and brain aging in general, and how the evolution of that is fitting into research on aging in general. Key thing that drives that is that in the 20th century, understanding of most of the illnesses of life came ultimately down to knowing what was inside cells and inside tissue, and that was really driven by pathologists for the entire 20th century. When it comes to the brain, people don't like needles put in their brain. So it's a major problem to deal with moving an understanding of brain disease forward at the same rate it can go forward in conditions that have to, hematologic conditions, kidney conditions, or almost anything else. And it puts us in a peculiar cultural setting in terms of how the field is going forward. What did I do? So, as you know, Alzheimer's disease began with descriptions by Alzheimer at the beginning of the 20th century. He was a neurologist and also did neuropathology, and he described his first patient, this one, Auguste Deter, early on, and in describing this, he pointed out to his colleagues that it was very peculiar because if she had been a decade older, he wouldn't really have paid any attention to her. She would have been just considered to be senescent, and of course, forever and ever and ever, we've known that getting senescent is as normal as anything gets, just like dying is normal. What he then did, when he started describing this condition, is he pointed out this is peculiar because it looks like the only thing wrong with this person is that her brain is aging too fast. And when he looked in her brain pathologically, he saw what he'd seen in much older people, most of whom had had senility, senescence at that point, which were essentially tangles and plaques. This was an exciting thing for the other physicians in Europe at that time because, as in most of the 20th century, we in medicine think of disease as a result of something external, some insult, some injury that is acting upon us and somehow changing us. So this idea was, he was putting forth the idea that this is a totally normal process that is moving at a different rate. However, fairly soon in the 20th century, it was realized that there were families that had this. What that then did, it allowed the physicians to say, oh, this is just like all the other illnesses we see. There is an assault, an injury, something else that produces this. It's just that it's a genetic injury, a gene somehow to our inheritance. So that was comforting, basically, to the people. And that idea remained. Now, when I was in medical school, we were not allowed to make a diagnosis of Alzheimer's disease for anybody over 65, 65 or over. The people who'd been studying familial Alzheimer's disease at that point knew that most of the lesions they saw in the brains of those patients looked like the ones they saw in old people with what they were calling a variety of different things, but senility, senescence, organic brain disease, a variety of other conditions. And they said, you know, if he developed this on a day before his 65th birthday, he would have this disease. The next day he wouldn't. So obviously that made no sense at all. So a number of physicians began pointing this out. Bob Katzman, Bob Terry, pathologist, and another number of people who moved from New York to San Diego were the ones who made this, you know, this interesting observation about that. I don't know what I did. Did I just turn it off? Probably sticking my thumb on the wrong thing.
Our normal helper is out.
I see. So anyway, this was an interesting thing that again, Bob Terry and Bob Katzman, so Terry was a pathologist, Katzman was the clinician, so it's showing here, but it's not coming up there. So they said this doesn't make any sense at all. We call it Alzheimer's disease when they're 64. It's clearly the same disease when they're 65. But what this did is it moved this condition from being a very rare condition to being one of the most common we see. And in those early papers, they talked about it being perhaps the 5th leading cause of death, or more than that, and was of the very most major importance in terms of public health of the nation. And that began to happen around 1968, 69, 70. There was a movement getting started just at that point to establish an institute on aging. Any ideas at all?
The projector is not working. My thought would be
just
if
to
you
use
keep
that.
talking and I'll turn that off and start it again. We'll see what happens.
Okay. So at that point, there'd been a book written by Bob Butler, who was a psychiatrist in New York, called Why Survive, which got a Pulitzer Prize. And it was really about the nature of aging and how this ought to be a serious health issue. There were also interesting definitions of what makes a disease. The only one that I've ever heard that I agree with is a disease is anything we want doctors to try to fix. Anything that we choose culturally to be placed in a medical challenge, we can call a disease, whether it's crooked teeth or whatever it is. So they basically said, we really ought to, I'll just do that. We really ought to make a major effort to do this. So the National Institute on Aging was founded with the idea, what did I just do? Did I just something again? Maybe so. Yeah. Anyway, in 1974, Congress established the authorizing legislation for the National Institute on Aging, and immediately it was identified that Alzheimer's disease and brain aging was central to the purpose of the Aging Institute. Bob Butler, the first director, found Zovin Khatchatourian and gave Zovin the task of planning a multi-decade approach to trying to understand what brain aging and Alzheimer's disease really was. And Zovin decided the first thing we had to do was to make sure that everybody was talking about the same thing. I want to say that really began to happen in the early 1980s. So the first meetings about the diagnostic criteria to define what Alzheimer's disease was occurred in 1982, 1983. And that basically launched the whole field of Alzheimer's-related work that we're in now. So there have been about 35 years of fairly intensive research in that. Since that time, since 1983, in those years, there's been a great deal accomplished, almost all of which has to do with questions of metabolism, biochemistry, animal models, and that sort of thing. So a huge amount of progress has been made. There was a recognition right from the very first that the amyloid plaques seen in Alzheimer's disease had to have a central role, as well as the neurofibrillary tangles later shown to be associated with phosphorylated tau being involved in that as well. It's been shown, of course, that there's a relationship between the accumulation of A-beta in the brain and the loss of neurons. And there was an identification in the later 1980s of the APP protein that is then shared and gives rise to A-beta. There was clear evidence that familial AD was often related to specific genetic abnormalities that involved that molecule. There has also been a great deal of work on the apolipoprotein 3 molecule and recognition that the three most common alleles clearly distribute themselves differently. On the other hand, whereas there's been a huge amount of progress in the laboratory kinds of studies, there's truly been more failures than not in the clinical issues. So we really do not understand precisely the development of amyloid plaques and neurofibrillary tangles and truly how those are related to the development of the clinical disease. The only really strong risk factor, the apolipoprotein E alleles, we have no clear understanding of why there's a difference between 2, 3, and 4 in those as well. We really don't know why plaques and tangles are so clearly related, but they're almost perfectly related in the standard clinical Alzheimer's disease case that we see. Epidemiologists are always looking for risk factors to guide prevention, and really the only two that have been consistently reported are apolipoprotein E4 and advanced age. So it's sort of embarrassing that in 30 plus years there has been really only that kind of research understanding the clinical illness. In addition to that, in the last few years there's been a huge amount of money spent on clinical trials aimed at prevention, a large percentage of which were with the idea that if we could block the accumulation of amyloid in the brain, people just wouldn't get demented. Essentially all of those have failed, and it's not clear why they've failed. Now in the beginning of this, when Zovin Khatchatourian had his meetings, they brought together people to be sure we were using the words in the same way, and the first issue was how will we define clinical Alzheimer's disease. Guy McCann from Boston and a group put together a very basic set of criteria to define what Alzheimer's disease was. But they made it clear that the best you could do was to say, if people meet these criteria, and they were very non-specific, gradual onset, gradual progression, starting with memory problems and progressing to multiple other domains, you could say those people had probable Alzheimer's disease. But the group made it very clear that was not definite Alzheimer's disease. It was essentially a temporary diagnosis put on those people that was called probable. In order to make a definite diagnosis of Alzheimer's disease, you needed to see the tangles in the plaques of the brain. So you couldn't make a diagnosis without looking at tissue from the brain. This then immediately said we need the pathologist to give us diagnostic criteria for neuropathology. The very beginning of this, Khachaturian, Melvin Ball, and a number of other people really put forward the idea that the key thing you needed to see was amyloid plaques. They didn't even really clearly distinguish between the neuritic plaques and the more soluble plaques that are around them, but any sort of plaques. And it became quite clear that that wasn't enough very soon. And about a decade later, a group of the pathologists got together and decided, and this was the CRAD group, there were specific criteria. You had to have a specific number of these plaques in the brain to see them. But it remained almost entirely related to how many amyloid plaques you would see. Around 1996, Brock and Brock pointed out that perhaps even more strongly associated with cognitive impairment than amyloid were the tangles. And he looked at thousands of brains starting from the cord up into the brain and saw what he took to be a gradual progression of the regions where the tangles were seen, as though something was growing upward into the brain across those things. Very quickly, the field sort of said that's a cleaner set of criteria to meet. Specifically, an area, the occipital cortex and the striate area, if you saw even a single tangle in that area, it was generally considered enough to be the high Brock stage, which really was plenty good enough. So as the Brock stage was Brock 0 through 6, and a 6 and a 5, almost everybody agreed, yes, that's enough to attribute the cognitive impairment to. And a 4 was sort of, and below a 4 just seemed to accumulate in people as they age very commonly, but attribution of the loss of cognition below that stage was really not suggested. Now, when this was happening, the reason for doing these criteria was less to understand the pathogenesis than it was to do with what pathologists are often expected to do, which was just simply tell the clinicians, did you get it right or did you get it wrong? So what that meant was, as long as you had criteria that were working criteria that served this purpose, you didn't really have to go too much further than that in understanding the basic biology.
It's not fixable now.
Okay. So I have some pictures here you can't see of tangles and plaques. I could do that, yeah. Well, it's probably too hard to see, but
And it will be available for later viewing on the departmental website. It's being recorded, but it's not being shown.
so this is a silver stain of a neuritic plaque with the neurites easily available. And these are tangles, flame-like structures in the cytoplasm of neurons, where plaques are accumulation outside of neurons. When this was happening, in the mid-1980s up until the early 1984s, there was a whole spate of reports from various centers seeing patients with Alzheimer's disease clinically that basically said, we're really smart, we always get it right. Our pathologist tells us that of the people we say have Alzheimer's disease, yes, we got it right. But what they really meant was the pathologists were applying the criteria that had been lifted from the study of familial Alzheimer's disease, which was basically if you see even a fairly moderate number of plaques, that's all you need. So it wasn't much more than that. The other thing that they made, which is not like what epidemiologists are trained to do, is that in comparing those brains, they chose really healthy people to be sure they were not demented. So it was not a fair comparison. These were, people who were obviously sick compared with people who were obviously not at all sick. So there were some problems with those early definitions that were really quite severe. The other thing is they said, if you see the plaques, you obviously look to see if they've had infarcts. Everybody knew that some people have a stroke, then after the stroke they began to lose cognitive functioning. So it was assumed that there was a stroke-related dementia, and you wanted to be sure you didn't have that. And in Parkinson's disease, it was recognized that, yeah, some people with Parkinson's disease lost cognitive functioning after. So they would look for Lewy bodies, they would look for infarcts. The infarcts they looked for were largely the large infarcts, which were the cause of strokes. Now, as an epidemiologist, a stroke is not an infarct. A stroke is a clinical event that is attributed to a loss of a substantial area of their brain because of vascular changes. The lacunar infarcts, much smaller and in other parts of the brain, but there hadn't really been much of a relationship thought through between the lacunar infarcts, for example, in the deep gray nuclei. So it was really, they were really looking early for big strokes and didn't really see much. Between about 1992 and 2010, there were a couple of really major changes that have completely changed the culture of understanding these. The first of those was there was a decision made with funding from the NIA that understanding this disease would require more than simply looking at the patients who came into clinics and to see their doctors, and more than just comparing those with healthy people. So it was recognized that what we needed to do was to understand, for the sake of the public health of the nation, the frequencies and correlates of the dementia cases called Alzheimer's disease that developed within communities. So at that point, there were 3 fairly large studies that were established almost at the same time. The NUN study here, the Honolulu Aging Study in Honolulu, and the Religious Order Study. So within 2 or 3 years, 2 of those were actually run and operated mostly by Bill Marksberry, who's a wonderful man, good friend of mine, who helped me get the study started in Honolulu. And he was the one who basically did all the heavy lifting in pathology with the NUN study, and then worked with me to put together a way to approach neuropathology in Honolulu, and was responsible for all of those. The cool thing about that is that means they were really looked at by one person conceptually on how to do this, and that one person sort of defined how you define words and things. So Bill was an enormous important person in that. In the early 1990s, there were a number of other things that happened. There was a recognition that you could see Lewy bodies in the substantia nigra and in the locus with an H&E stain pretty well. But if you looked at them harder, there was a much better molecule to look for, alpha-synuclein. And my understanding is that there were people with antibodies to alpha-synuclein who were studying the songbirds, canaries and other things, and they loaned the antibody and saw these beautiful. And then with those antibodies, you could see alpha-synuclein positive inclusions in a lot of other places in the brain. In the first cranial nerve, and certainly in other places. So that moved, and it was recognized then that the people who'd been recognized as having extrapyramidal disease, but also dementia, a lot of them had Lewy bodies not just in the substantia nigra, not just in the pigmented brainstem nuclei, but also in the neocortex. Because of Bill Marksberry's efforts, I had asked Bill when we started doing a study that we were going to be looking for things that pathologists had not yet recognized. So I asked Bill, from his experience, to make a record of everything he thinks is weird and unexplained that he sees in the brains of old people, put that together in a form. Among the things were he wrote down microinfarcts. And I asked, so Bill, what's a microinfarct? And he said it's just a little area where you've lost neurons, and there's gliosis. We don't know really what causes them. We assume it's vascular. But they don't seem to do anything, and nobody in their right mind could think that they could cause any trouble because they're so few and far apart and in all kinds of different places. But we put it on the form. And as an epidemiologist, I got compulsive when we put together a form with a lot of different variables, guided by Bill's judgment and experience, looking for microinfarcts. So when the data came up, I did what people would ordinarily do with infarcts. Is there a location of this lesion that you might expect would be associated? But the problem is cognition is different. The nature of dementia is that it is a process that involves all aspects of cognition eventually, which seems to me, obviously tells you, it ultimately involves all areas of brain to some extent. It's not going to stay focal. So if a person has even a big stroke, a big infarct, they don't necessarily have real cognitive loss that way at all. So focal lesions obviously don't ordinarily produce dementia. If you have dementia, it implies that there is a distributed process that impairs the function of neurons and their intercommunications. So what I was able to do was just count them. So there'd be one here. We looked at about 18 sections. There'd be one here, one there. By the time we counted 3 across 18 sections, the odds ratio for a relationship with dementia was as strong as a Brock stage of 5. So what that says is that there is a distributed process marked by widely distributed microinfarcts which produces dementia, and that pretty much fits what we were looking for vascular dementia. Prior to that point, pathologists had defined to come up with the vascular dementia criteria, because if you start with strokes, most people with strokes don't become demented. So there's no clear way to do that. So the microinfarcts allowed us to put forward a set of structural changes which seemed to be quite specific. So we were working toward vascular dementia at that point. At the same time, Bill and Darren Davis, who was working with them at Kentucky, they were writing down this weird thing called hippocampal sclerosis. And I asked Bill, what is hippocampal sclerosis? And we talked about seizure disorders and those sorts of things. And I told him, there are a lot of these. And he said, no, it can't be. Went back and looked, and he came back and said, you're absolutely right. And that has obviously developed to the hippocampal sclerosis of aging. At that point, there are only a couple of other people who have recognized that there was something there. So now what we're doing is we're moving forward in terms of the changes in the brain, which seem to be independently associated with a loss of cognitive functioning. And again, diffuse loss of cognitive functioning, recognizing that this means some sort of generalized brain pathology. I also began to recognize that the loss of brain tissue, and Alzheimer himself, and everybody has recognized that brains get shrunken. And a little bit of shrinkage occurs in people who are not cognitively impaired all the time. Most people who have Alzheimer lesions, and it turns out most people who have a lot of microinfarcts have a lot of atrophy. Now there's a problem with atrophy, and that is because most of the modern trained neuropathologists that I talk to don't like touching dead bodies. And what they want to do is have the diener take the brain out, prepare it for them to look at. When you take it out of the head, you can't see how big the hole in the head was for the calvarium to fit it into. Now, if you atrophy, what's going to happen is the brain is going to shrink down and leave that space, obviously, around it. So if you know the intracranial volume and you know the volume and mass of the brain, you can impute how much has been lost. That's atrophy. The trouble is that very few people, now, imaging people do this all the time. Right from the very beginning, they recognized that the strongest correlate of hippocampal volume was your intracranial volume. That was the strongest no matter what. So initially they were bitemporal measurements or a variety of things, but that field evolved really forward. And of course, because the neuroimaging people are so sensitive to volume changes, to tell you what's been going on regionally and generalized, they're very good at recognizing atrophy. Pathologists aren't. And it's something that really has to change. Now, there's no promise that all atrophy you see in dementia is driven by those nasty A-beta molecules or by the microinfarcts. There may very well be separate mechanisms that are responsible for loss of gray matter and white matter. And if we knew what they were, and if they are truly independent of the others, that gives us another target for prevention. So one of the things that pathologists need to do is to figure out how to compete with neuroimaging people to address the issue of generalized brain atrophy. And I think it's a crucial thing that has to be done. It may be as simple as when you work with your technicians, getting them to measure the intracranial volume with rulers. And if you could then impute the intracranial volume from a series of 4 measurements, you'd be able to compete with neuroimaging people in getting at true atrophy as opposed to simply the brain size. Really crucial issue. So there's a bunch of pictures. Picture of hippocampal sclerosis, Lewy bodies. Now a microinfarct, what our pathologists do, and Bill Merksbury was the first one who did this, he just scanned over his slides on low power, and his eye was so good, and every pathologist I've worked with is able to do this. Spot an area where the neurons have dropped out, and there's some evidence of gliosis, and that's enough. Now, what's interesting is we do 8 micron sections, but these are not 8 microns across. About the smallest are about 50 microns across, up to 300 or 400. But you know, when those sections are cut, it's a random chance where you're going to cut it relative to the microinfarct. And it'd be really interesting to know what the distribution of those were. We don't know if there's a cystic center of most microinfarcts or not, because we don't look for it. We just see them in whatever 8 micron sections we look at. Anyway, that's a microinfarct. Okay, so at this point, in the Haas data, there were really 5 different kinds of lesions we've seen. We had seen, and I began to recognize, as did other people, they're really not tightly associated with one another. Atrophy is pretty well associated with the Alzheimer's lesions, but not entirely. There is atrophy, which is associated with dementia, which seems to have nothing to do with plaques or tangles. There are people with plaques and tangles who have very little atrophy. They tend to be less cognitively impaired, which makes sense. If our thinking requires neurons to exist and talk to one another, you would think that that's what you would get. And that most cases that have plaque and tangle disease and are demented are probably losing or have lost neurons. Again, we're thinking 3-dimensionally and actually 4-dimensionally, with time as the 4th dimension, and 5th dimensionally if we're thinking about the chemicals that are going on there as well. But we really mostly only look at things 2-dimensionally, pathologically, even though we're thinking in terms of those dimensions. Anyway, in Haas, about 30% of the people who died, their average age of death was 89. About 30% of them have moderate or high levels of plaques and tangles together. And for tangles, it's a Brock stage 5 or 6. Almost exactly the same amount in Haas had microinfarcts, and 3 or more across 18 sections was what I called a moderate or high level. Now the nuns have substantially fewer than that. Very interesting observation. When I asked the pathologists, these are little itty-bitty things. Doesn't the brain tissue collapse around it and don't they disappear? They all told me no. Strokes, infarcts never go away. They're forever. That doesn't seem to me to make a lot of sense, because throughout your body, things are changed all the time, and you would guess that at minimum, they got smaller. And if you're taking randomly selected 8-micron samples, as microinfarcts mature, they ought to disappear, or at least they ought to get fewer in number. Now, what's different about the nuns? If it turns out that the nuns accumulate microinfarcts toward the end of their lives more slowly, that would be enough. So in the guys in Honolulu whose lives are probably a lot more pressured than the nuns, they may continue to develop microinfarcts toward the end of their life. Now, what this basically says is what we really lack in pathology is temporally longitudinal data. We're only going to do, but we have to think about everything we see in terms of temporal sequence. That's essential to really understanding what's going on. The difference between nuns and the guys in Honolulu is going to be something important. The nuns also have somewhat fewer lacunar infarcts, which are quite closely related in frequency to microinfarcts. So microinfarcts are associated with large infarcts in numbers, much more strongly associated with lacunar, but they're actually more frequent and more tightly associated with dementia than either of the other kinds. Now, the neuroimaging people are always telling us they know what causes vascular dementia. It's that funny distribution of water giving you white matter changes, and that's probably going to be due to small vessel disease. They're probably right. And it may very well be that microinfarcts are that as well. Now, the old-time pathologists, when I talked to them about, when you say a brain has atherosclerosis, what do you mean? They told me, when you cut the brain and look at it, and you look at the little arteries that you can see, they've gotten weird because they're all curly and bent around and tortuous. When they're tortuous, the blood that's going through them is obviously losing energy. So the potential reason that microinfarcts occur is not because of an embolus, not because of anything like that, but it may be just because of a loss of perfusion pressures delivering oxygen and glucose to specific regions of the brain. When I was in medical school, the neurosurgery department in Seattle had this spectacular machine, which was X-ray cinematography. When I was on neurosurgery, we did this service where a guy was thought to be bleeding from a little aneurysm up in his brain, and the surgeons were going to go in and look at it. And this was pre-CT, pre-MRI data. So what we decided we had to tell the surgeons was where it was. And I and the resident basically put a cannula into a carotid artery on the side we thought it was, got a vial of hypate or whatever we used, a contrast material. And when he told me push, I pushed it in, went up into the vessels, and we took frames of this, like at 60 frames a second, X-ray. When we were all the way through, we went back and looked at the pictures. So this was really very high performance, essentially, photography. What I saw amazed me because these little arteries inside the brain, as the dye went up into it, started jumping and wiggling like snakes and worms inside the guy's brain. Well, thank you very much.
Be careful what you put.
Yeah, absolutely. So I think what's happening is that there's a structural change that's very common in the small arteries in the brain, and what they do is they produce a tortuosity. Now, why would that happen? Well, what we do know happens is that the amount of collagen 3 and collagen 5 that you see in the walls of these vessels goes up. There's clearly a change in compliance. The internal elastic lamina, which are in those, basically is continuously replaced by protoelastin being brought out into the artery walls and placed in place. But if there is a microscopic molecular tear, because simply of the trauma of having pulses of blood pushed through those vessels, the way it'll get fixed is with collagen band-aids. And collagen is not elastic. what that would do is it would decrease the compliance generally. So then you got these pulses of blood coming into the brain in big pushes every time the heart beats, and the blood has to go somewhere. If the artery can't expand enough so that the cross-sectional area is sufficient to hold the extra volume, what will it do? And the answer is it'll stretch. It'll stretch and lengthwise and get longer. So these tortuous arteries are in fact long, which means that between pulses, it may sort of flatten out, like a birthday toy you blow into instead. So what I think we're seeing is the Virchow-Robin spaces around arteries are the result of these changes, loss of compliance, and those changes. And what's happening then is that by the time the blood gets through that, up into areas of the neurons where the neurons are waiting for oxygen and glucose, the perfusion pressure is down and some of them die. Now, if the good astrocytes are doing what they're supposed to be doing, maybe delivering some lactate and pyruvate, those neurons may last longer until it's inadequate. So my guess is that as microinfarcts are developing, you begin to get injury to the neurons associated with a change in metabolic integrity, which the good astrocytes can't quite fix, till eventually the neurons start dying, and that's when we see. So, microinfarcts don't do it. They are a marker for diffuse ischemia and a diffuse decrement in the efficiency of metabolism. That's my own personal view. I asked Bill Marksbury about hippocampal sclerosis. He said he had no idea really what was doing it. But we saw that essentially half of the cases were strongly associated with Alzheimer's lesions, and the other half weren't. Now we know that hippocampal sclerosis is associated with other kinds of things, 2 or 3 other kinds of changes, including frontotemporal dementia and some other sorts of things. So it's not clear that hippocampal sclerosis is one thing. What's clear is that the frequency of hippocampal sclerosis goes up more steeply with age than almost anything else. So something's happening. Pete Nelson and some other people have suggested that this is maybe a vascular condition. I doubt that it is, but we don't know what it is really what's driving it. And then I am so impressed that we need to get our act together to understand why neurons die diffusely, and its relationship to white matter loss. What part of it is exclusively related to amyloid and tau, and what part of it isn't. When we understand that, we have another target for intervention. So we don't just have to prevent plaques and tangles or other things. If we could identify specifically how to preserve the neurons to whatever it does, it would help us a great deal. So anyway, at that point, when I was doing these, we really had 5 major kinds of neuropathologic abnormalities in the mid-1990s. And that was real progress, because in 4 or 5 years, we'd gone really from having one plaque and tangle and tau thing. Now we've got 5 things. And it was pretty clear that they were pretty much independent. These were not all first cousins of some underlying process. They were really independent. So the first thing was then to look at the frequencies, which I just showed you, and then to say if you looked at them one at a time, defining levels of moderate and severe levels, are they really similar or dissimilar in how tightly related to dementia are? And the answer is every one of these 5 can do it to you the same way as plaque and tangle process, whatever that marks, can do. So once again, these are all powerful, independent causes of dementia. Now, there's been a major change which I alluded to, which that there's a lot of different lesions, each of at least 5 different kinds of lesions. Now with TDP-43 and a couple of others, more than those things. TDP-43 is at least partially independent of those other sorts of things, and there's probably a substantial overlap as well. There have been a series of papers And essentially all of the papers that show this are not from sites where people come because of dementia. There are sites where they've gone into the population and said, in the population, what do we see? We follow them forward longitudinally, what do we see? So the answer is, there have now been a host of papers which have absolutely demonstrated without change across them that as you get old in populations, these 5 or 6 kinds of lesions all come forward, and all of them are independently associated. There's no question about that. Now, what does this mean? What are the inferences we take from those data? Well, first of all, since the dementia that these are associated is pretty much indistinguishable from most cases of Alzheimer's disease, and because the clinicians have for the most called them probable Alzheimer's disease, what that probably means is that cases that come to sitters for study with probable Alzheimer's disease as the diagnosis, at minimum, are markedly heterogeneous. They are not driven by a single process. They're driven by at least 5 separate, distinct processes, each of which is independent to some extent from the others. The other thing is that if you look at people who are not demented at all, they have lesions too, just not quite as many, and in a collective sense, not as many combinations, which turns out to be the key issue here as an epidemiologist. The other thing is, everybody knows the risk factors for Alzheimer's disease are advancing age and the ApoE4 allele. Everybody knows that if you do the age-specific prevalence of Alzheimer's disease or dementia in general, it doubles about every 5 to 7 years. So from age 60 it goes to 1 to 2 to 4 to 8 to 16 to 32, wildly like that. But if you take the age-specific prevalence of each one of these processes marked by its own lesion, it does not do that. The age-specific prevalence increase with age is much, much, much flatter. If you add up how many different things you have, that does double every 5 years. So what we're doing is we are seeing a physiologic phenomenon that is related to the relationship among different processes and how those add together. At my age, I've begun to recognize that the key thing about aging is Murphy's Law. Everything that can go wrong is gradually going wrong, which means that the frequency of having more than one thing doubles every 5 or 6 years. If you look at the risk of dying, it doubles every 5 or 6 years. So what I've just described is a fundamental characteristic of aging within an organism. So this is multi-system failure, and within each system, failure of different components. Fundamental principle of the diseases of aging. Don't forget it. It's going to apply to MIs, it's going to apply to lung disease, it's going to apply to presbycusis. Everything you look at is ultimately going to be, we're losing function and structure because system failure, because Murphy's Law has taken over. Occam's razor works for adults in the middle of their life. It doesn't work when you're getting older. I did it again, didn't I? Nope. Now, I'd like to tell you about, of the list of papers that I described for you, it went off again, didn't it? I did it? Okay. I'm sure I'm doing that. Apologize.
No, they said it's a common problem, and the room's being remodeled, but it can't happen until March.
We can just stand here.
I know. That's what the projector's getting.
So anyway, there's this really interesting article published less than a month ago. So you can't get a full copy of it yet. From the Religious Order Studies in Chicago. First author is Patricia Boyle. Julie Schneider is this pathologist in this, and she presented it in London at last year's Alzheimer's meeting. And basically, they began to look at this process of all these different combinations of these weird different kinds of individual pathologies. And the first thing they did is they took each pathology and changed it to a score of yes, no. Not it or you don't got it. So every one of them gets a 0 or a 1. And then they looked at all possible combinations, probably temporary permutations as well, but certainly. And in their data, they saw over 200. This means that if we're defining an underlying disease system in aging responsible for probable Alzheimer's disease, they're saying there are more than 200. That doesn't make much sense. But that's the view. Now, the failure of Patricia's paper, I think, is that she didn't really take into account. She called them 0, 1, if they're present or not. But like all kinds of insults that we see, it's not just if you've got it or not, it's how much it is, where it is, and its relationship with other things that reflect system So the way that we looked at this in the study that from Hawes and the Nunn study together that we published a little less than 2 years ago, we looked at all these in the same way in the two studies. Again, because of Bill Marksbury's contributions, we could do this. They were very similar in the two studies, the Nunn study and the Hart study. And what we decided to do was to try to identify the major underlying processes linked to each one of those. And when I've done most of this work, these are the 5. So there's cortical Lewy bodies, hippocampal sclerosis, amyloid plaques, and tangles, atrophy, micro and lacunar infarcts. And the key thing is that microinfarcts and amyloid lesions clearly drive a lot of atrophy, but not all of it. And the others are pretty much independent. Clearly, something of the Alzheimer's process is driving some part of hippocampal sclerosis as well. But precisely how, I don't think is yet clear. Now, what we decided to do is say, maybe your risk of getting demented is less related to the bad luck of getting type 1, 2, 3, 4, or 5 than the bad luck of having Murphy's Law act on you, and the amount of different elements that you need to keep the whole system of brain function going. Maybe the number is what it's at. So now we're into the straw that finally breaks the camel's back. Brain is so redundant that if you have multiple ways of solving problems, until you've run out of ways, you may slow down, but you may keep qualitatively your functioning. So what we decided to do was to come up with measures of moderate and severe pathology for each one of the 5 lesions. And I elected to scale them as 0.4 for moderate and 1 as severe, for the simple idea that if you had 2 moderate, that would up to 0.8, and you could compare it with a 1, which would be for one singular one in severe. So what we did was we generated for both studies a total neuropathology index by adding up across the 5 lesions that I was working with at that point. The scores 0.4 or 1 for all of them. This is what you see. This is a score along this way. The height is the risk that you were demented. It is mind-blowing that as you increase the total number, it doesn't make any difference which one it is. It's just the total burden, which is not a surprise. If you're looking at a system failure, anything that makes the system failure may be as bad as anything else. So it's dramatic. And this is the same thing. This is in Hawes. The previous one was in the Nunn study. So this is really strong evidence that as beautiful as our histopathologic stains are, in understanding pathogenesis of dementia, it may be a system failure for which you do not need the aesthetic beauty of stains if you simply understand the total loss of structure and function that occurs. So that's the idea. It's nothing more than an idea at this point, but that's the idea. Now, in the Haas, then I pulled out 252 demented people and 251 normal people and said, okay, what is the distribution in those two groups of this total burden? So 0 meant that the person didn't have any lesions at all that we could see. In the people who were demented, it was 4%. That means 96% of the dementia can be attributed to structural brain lesions. You don't have to worry about depression. You don't have to worry about those other kinds of things. It's all due to structural lesions that a pathologist can see. A big chunk, 11%, was associated with a single type of lesion in moderate level. Most people with moderate levels of a one kind of thing were not demented, but the risk does go up. So what it really says, looking at the demented, most of them had 2 lesions or close to 2 lesions to get severely demented. If you look at the normal people, what you see is that 28% had no lesions at all, but there were a bunch of people with some lesions. So again, this is how I feel when I get up in the morning. I can feel myself aging. My joints are giving me trouble. My hearing is giving me trouble, but I'm still functioning, still managing to get grants. So here's the principle. It's that the increasing neuropath burden appears to be the cardinal pathogenic finding that's linked to dementia. Dementia generally, which really for most people means probable Alzheimer's disease. And that is rather than blaming it on any single type of lesion. The other thing that's really cool is that as an epidemiologist, we use logistic models all the time. There are logistic models because we measure the effect as a logit, which is essentially an exponent. So the models start with the basic level, and then you add the influence of each other variable in the model we're looking at. And you measure the influence with an exponent. What happens when you add exponents? Adding an exponent is multiplying the arithmetic equivalent. What that means is if you have one person with a lesion that gives you an odds ratio for dementia of 3, and they have another lesion that also gives you an odds ratio of 3 by itself, and a third one, you would expect an increased risk of 27-fold. That's what exponents do. These are the data looking at people. So the people who, the reference group here is they had no lesions. So they have an odds ratio of 1. Then if I group them by an index of 0.4 to 0.8, so these are 1 or 2 moderate level, 1 to 1.8, 2 to 2.4, what you see is that there's an exponential increase in each one of them. These are individual groups. They're not the same people. So the person that has more of these, eventually the estimate was an odds ratio of 99.
Wow.
If you had more than that. And that was in the Nun study, and in the Haas, it's about 40. Now the numbers get really rough. So this just really tells you that it is not a small matter to have more than one thing. So then again, this is the camel's back straw. So if you're going to look at the prevention of dementia, that's what we have to pay attention to. I'm getting close to my time. I wanted to tell you about the revised criteria. Pathologists began to see this, and they basically, Tom Montine and Brad Hyman and a group of about 30 other pathologists got together, as I'm sure you guys all know, and they came up with criteria for the diagnosis of Alzheimer's disease. The first thing they did is they bit the bullet and said, we're going to define Alzheimer's disease not as a clinical illness. We're going to define it as the underlying disease process. Now, everything else in medicine works that way. Osteoporosis, arthritis. We understand that the disease we're trying to prevent is not just the endpoint, not just the phenotype, it's the whole thing. But pathologists doing criteria for dementia had not previously done that. So they said, we're going to say you have Alzheimer's disease as a process, even if you're very bright. As long as in your brain we see this. But also, because attribution of the relationship between pathology and clinical is really important, you better take account of the other major lesions. So their criteria requires that as well. But this has caused trouble. And it's reason we now can't, now neuroimaging people and clinicians have their own cottage industry. And to get them to define the same term different ways will often disrupt their grants. It gets to be a heavy-duty thing to talk about what Alzheimer's disease is. So if Alzheimer's disease is this underlying process, and we say the key parts of it have to do with amyloid and tau, then that's something different than the neuroimaging people, the geneticists, and the others throw together in groups when they look for the genetic causes or the characteristic brain volume changes. They're now looking at two different groups of people. And we are on the horns of that dilemma right now because we're talking a different language. And it's important that that be figured out. When we look at this, it looks like only about a quarter of the cases that clinicians send to us for genetic studies and neuroimaging as probable Alzheimer's disease, only about a quarter truly have just Alzheimer's lesions. Another quarter have Alzheimer's lesions, but with other stuff. And about 40 to 50% don't have significant amounts of either of those. So about half the cases that for the last 30 years have been put into series of Alzheimer's cases were wrong. That these were misdiagnoses because of a lack of appreciation of what pathologists had been unable to tell them because we don't do brain biopsies. We only could see what's in the autopsy. And most people don't like knowing what's in the autopsy anyway if they don't have to. So it's a problem that is where we're at. Where do we go from here? The answer is we need to be able to, we meaning the medical community, recognize the changes in the brain that our pathologists see while people are still alive. At this point, the only way we can do that is with histopathology. Nothing serious is going to happen in the field with documented effective prevention until we can identify clearly, specifically, and comprehensively the brain lesions that we know are there in clinical cases. That's all I want to say. Thank you.
Thank you, Lon. That was a wonderful, eye-opening talk, and yeah, have a couple minutes for some questions.
Are you saying that the statistical independence of your 5 lesions rules out the possibility of common cause for all?
I'd never say that, but it sure looks like it mostly does. Atrophy is the most important thing to us to study, and because you don't do histopathology for atrophy, nobody's very interested in studying it among pathologists.
But what about like sampling error, for example? Suppose something is there, but you don't see it under the microscope because you've just been unlucky in your.
If it were equally there in the different kinds of lesions, we ought to see it, it seems to me, in frequencies. And we don't seem to. But you're right. What we ought to be doing now is asking that question and figuring out a way to answer it. But my own view is we've assumed since 1982 that if we understood amyloid, I remember Zavin saying if we could just have the rates of amyloid accumulation, we'd have the total rates of dementia reduced. They'd have that. It's not true. But I think those are the questions we have to answer. And this moves us into molecular and cellular physiology and pathophysiology, into system failure biology. And we are entering a phase of medical research where we have math specialists in our departments, and those people may be able to help us.
Any other questions?
Just a comment. I was intrigued by the tortuous blood vessels you demonstrated. There was a fellow here, a mechanical engineer named Harry Blackshear, who's very famous National Academy of Sciences, and he worked on atherosclerosis. And I remember, you do these studies in animals, and it's so critical to have clean and clear laminar flow along the vessel walls.
Absolutely.
And at every place there's shear, that's where atherosclerotic lesions became. So at bifurcations, and so you get turbulence. So you could see sort of a downward spiral by virtue of having whatever it is, type 1 or 3 collagen, and who started that in the first place.
Right.
But it could just exacerbate.
This is so fascinating. And again, I think if we understand that, since a very large number of the cases of dementia are demented because they do have Alzheimer's lesions with relative ischemia. If we could deal with that, we'd have. Now, we did an interesting analysis. We looked at the circle of Willis. And so we looked at the vertebrals up to the basilar, a whole circle, and then the penetrating articles. And what we did with those is we did cross sections. Now, the trouble basically is the vessels collapse when they're formalin fixed. But if you measure the perimeter, you can make the assumption that the relationship between the perimeter and the maximum cross-sectional area during life is more or less constant. And when I looked at those things, and I asked which of those are associated with microinfarcts, the most important one was the size in the vascular. There were aneurysms. Now, the other thing, old-time pathologists, when you ask them, does a patient whose brain you held in your hand, did they have atherosclerosis? What a lot of them meant was they looked at the vessels coming into the circle. And what they really looked at were aneurysmal changes. They were fusiform changes. When you cut those sections, you don't see a lot of nasty cholesterol kinds of plaques. And when you go up into the small arteries, you don't see. There's some, but there's really not a lot. What they were calling atherosclerosis looks as though it's like the bellows for a fireplace. It looks like it's expanded, and it really looks like the basilar area is catching a lot of that pressure. So my bet is that tortuosity develops when you've exhausted the ability of downstream structures to take the extra volume to go in. All of that should be very accessible to clinical study and to research study. And I'm convinced that if we now, the other thing about this is what are the risk factors? The primary ones are midlife hypertension. Midlife hypertension presumably drives arteriosclerotic change, which I don't think is really cholesterol crud. I think it's a change largely in the internal elastic lamina and a replacement of the elastin, the protoelastin and elastin molecules with less elastic structures, and that gradually accumulates. I think the reason hypertension is so common to develop is our kidneys have these very sensitive systems for detecting a fall in pressure, and then it demands peripherally you give us more blood, so we're okay. So I think what we're really seeing is an evolutionarily explainable change in gradual accumulation of hypertension as demanded by the kidney. And if we ever want to change arteriosclerosis with renal disease and brain disease, we need to understand that that's a normal evolutionary mechanism. It has to be subverted with quick recognition of hypertension and treatment of it. Our estimates were that if we had effectively treated midlife hypertension, our total lifetime rates of dementia would decrease by about 25%.
All right, last question, and I think we have to hurry up because we went a little over with technical. Okay.
It's not possible using antibodies to remove the plaques, like with adalimumab. And I just wondered, have you modeled what the decrease in prevalence of dementia would be just by removing that one factor?
No, the only thing I've done like that is I have modeled what would happen if you had a preventive intervention that totally prevented the accumulation of amyloid. And it looks from two models, from Hawes and Manoa, you could decrease the lifetime frequency of dementia by somewhere around 30-35% if amyloid never accumulated. That's the closest I've done. I have not done that with trying to understand. You know, the problem basically is epidemiology is a very blunt instrument, and we can only deal with the numbers that pathologists give us. And we don't really have good numbers for those things. Thank you, Karen. That's really, really important.